I'm an independent consultant. I was told I need to be a good salesman for my business, so I am an independent consultant. However, it's my pleasure to work with fine companies like Regis who synthesize materials. So oftentimes I'll work with CMC people and work with chemical companies on elucidating the solid state physical chemistry and determining the relationships of different crystalline forms of materials during the process development phase of chemistry, of chemical development. So today's talk, basically the outline is I'm going to introduce you to polymorphism, the timing of studies that generally large pharma uses, having worked and compared myself with what I did in my time at Eli Lilly and Company, along with, we collaborated a lot with other companies, and much of what we do, seem to be in common, the timing and the like. So I'll discuss a little bit of the rough timing, the ICH guidance to polymorphism, discuss some of the analytical techniques briefly, many of which Regis has been adding to their work so that they can support the solid state activities more thoroughly. And then I'll talk about three kind of exceptional systems from a polymorphic standpoint. But in general, these are kind of exceptional systems. But in general, if you work hand in hand process chemistry with a person that's pretty knowledgeable with solid form relations, you can design processes that are very robust and can produce a single polymorph for product development and for the final product. And so that's what I'm going to talk about today. So polymorphism, there's a lot of different definitions, but the purest definition is that it's when you have molecules that arrange themselves in a crystalline fashion and in different ways such that they have different physical properties. And in the truest sense of polymorphism, they're chemically identical if one was to take and dissolve those substances and run solution in MR. So if you overcome the lattice and dissolve it, they're identical chemically, but they're distinctive physically in physical forms because of their interactions and the way that they pack themselves within the crystal lattice. And so polymorphism has a major impact on the physical form has a major impact on physical properties of the active pharmaceutical ingredient, but it can also influence a lot of the product dosage form's performance, many properties in the dosage form, the final dosage form, such things as solubility, particle size, shape, and morphology, water sorption and desorption, or roughly, people call it hygroscopicity, chemical and physical stability, so how much it degrades in the solid state, physically, whether or not it transforms to another solid state form, then the drying properties, purification, and yield, considerable, the actual physical form that you isolate has considerable impact on this. Flow and filterability commonly is impacted by the physical form. More so in the when you mix it with other excipients, the stability, the chemical stability can be impacted or differentiated. Two different polymorphs might have quite different chemical stability, which actually can kill projects. And then physical, it's important that we understand the physical forms and are able to control the physical form of our API, but even more so in the final product and throughout its shelf life to ensure that we deliver a consistent and stable product. It can impact the flow, blending, granulation, tableting, and many other properties that that are impactful in the actual process, the formulation process. dissolution. You should be able to differentiate polymorphs based on dissolution. And as the FDA puts a lot of stringence on having a robust method for discerning, they typically push you to have very, very tight controls on your dissolution testing, and you should, definitely polymorphism or even hydrate formation can have significant impact upon the dissolution behavior of your final product, and sometimes even bioavailability, depending upon the solubility properties, in the BCS class of your compound potentially. So I'm within the drug development process, the time that really the earlier the better, but historically my work has occurred with the salt form and selection occurs in early lead optimization at many of the large pharma companies. So when you're down to maybe 5 compounds in a certain program, say doing 4-day tox, typically for me, there would be about 5 compounds or 5 grams of each one of those compounds would be produced. It was important enough to the company to provide a couple of grams for salt selection so that we could ensure that at least the data that we would get in our early tox, 4-day rodent studies, that the bioavailability that we would get would be, or the exposures that we'd get in the animals would be somewhat relevant. And so in particular, crystalline versus amorphous, we'd want to make sure that it was crystalline material so that, or at least know what form was dosed. But during this period, with the 2 grams of material would be committed to our salt screening, and we would also, they work hand in hand. When you select the salt, oftentimes you find you're trying to crystallize it, and you'll find multiple crystalline forms during that time period. But then the other 3, so 5 grams would be produced, 2 grams would go for salt screening, and then 3 would go for the toxin ADME testing. But then I'm going to focus mostly on the polymorphism, which goes hand in hand with the salt screening, but it usually follows. You'll initially select 1 or 2 or 3 salts, and then you'll try to generate as many forms of those salts as possible. What we would do is we'd work with that first 5 grams that was produced, then the company would try to scale up larger amounts of material for more extensive tox studies in a second species. And we'd try to inform not only what salt, but what polymorphic form would be produced for those studies. And those studies would be a shorter, but wouldn't be as long as a GLP tox study, but they would be informative from the fact that they would try to do some dose escalation in the animal and get some sort of idea, and actually would try to establish somewhat observe some toxicological effect. So you'd want to be able to establish essentially a margin of safety within that animal so that you would know so you can convey to the regulatory body when you filed your IND what sort of safety margins you would be operating in within your dosing regime in first human dose. Now, I would say 90% of the time, at that point, so there would be about 100 gram scale at this point where those tox studies would occur, but we'd select the polymorphic form. We'd make sure, primarily we'd be trying to find the most stable polymorph that we would hope to commercialize. And in doing so, I'd say probably 90% of the time we were able to be successful and not have to go back and change form. It saves a lot of repetition of studies. One of the things, however, that happens, there's only 100 grams here, and later, when you do formulation development, you're going to have to scale up and have larger amounts of material. And some of these bulk properties or the flowability of materials and the like really can't be well assessed until you get a little bit later into development, when your formulators start working with materials and the like. And so you might uncover certain things that you don't necessarily like about the polymorph or the form that the physical form that you've chosen. There's no perfect forms. You know, it's rare that you don't hear some feedback about this or that should be better about your compound. But generally, you can do a good job of selecting the final form pretty early on. And I'm talking, a year and a half before first human dose. But there's certain information you get post or after you've made that selection. And one important factor is this route selection. So impurity profiles can change, and it's one of the most important factors in determining crystallization outcome. And so because rel subs are the most similar to the drug molecule, they're more likely to interact with the growing crystal form and what forms nucleate. And so it's an extremely important factor. And so the sooner you get your route selected, the sooner you can be more confident that you have the physical form and that you've identified all of your polymorphs. So the sooner you pin down your chemistry, the better off you are. But a lot of that does continue, as processes are developed, as they're scaled up. And then the doses aren't really established until you start getting out into the phase 2 studies, as you refine the dose, the efficacious dose. And then you start to get into formulations that are more representative of what you might make for your commercial product. And so you can learn, and periodically you may have to change. Like I said, less than 10% of the time, I would say, is when we would change forms beyond candidate selection. Part of the importance of the salt, the most traditional means of improving the bioavailability of a compound, if you have one that's in the BCS class 2 or one that's poorly soluble and you think that it might have to be a relatively high dose, is salt formation. And so this is why traditionally it's, salt versus free forms established very early on, and the solubility ratio of one versus the other can be as much as, a salt form can be as much as 1,000 times more soluble than, say, the free or the neutral form of the molecule. And so we try to, early on, that very first step is determining whether or not you need a salt, and determining whether or not you might have to enable it by making an amorphous form to take advantage of the enhanced solubility of the amorphous form over the crystalline form. But then once you've chosen that, you look at polymorphism, and also, about 30 to 40% of the compounds will form a hydrate. And so you almost always have to deal with hydrates, or a good percentage of the time you have to deal with a hydrate, and where that transition humidity or water activity occurs, and determine which one is best for your processing and for control of your product. But these are still significant differences, much less so in solubility ratio. But a hydrated form versus an anhydrate typically is about, the hydrate's about a quarter the solubility of the anhydrate in water. And one polymorph versus another, this nice paper by Abu Sarajudin from Bristol Myers Squibb in 2005, he took 81 different compounds and compared their solubility ratios to one another, and determined that it was a 1.76 on average fold difference. The metastable form would be 1.76 times more soluble than the stable crystalline form. And so those are significant. I mean, if you go too far into development and you have to do a form change, you're also likely going to have to adjust your dose, which means that your formulator is going to have to change its formulation, which means you're costing yourself a fair amount of money. And from my recollection, it was somewhere on the order of at least $500,000 to develop a good formulation for a tablet. And so it can get expensive. Early studies were typically done drug and capsule or with a very simple formulation. Later studies are done, in the final formulation. So you don't want to have to develop that final formulation, which costs a lot of money and time. So it's good to get your polymorph identified and defined very early. The ICH, there was a conference where they harmonized basically, and there were some issues actually in the late 80s with carbamazepine hydrate versus anhydrate form, where some generic manufacturers came out on the market and had a compound that would convert into a hydrated form, and the patients weren't getting efficacious doses from what I recall. And so it really brought the light on the importance of controlling polymorphism. Lilly, the company that I was blessed to work with for 30 years, was very, they were very well aware of this and controlled it. They had X-ray diffractometers clear back into the 50s. But some companies didn't have a full appreciation, and it wasn't widely accepted or understood within the pharma industry how important it actually could be. But some of those patients died with that carbamazepine, and so it heightened the FDA's awareness, and so the regulatory guidance came out in the mid-90s, partly because of carbamazepine, but it just as being good science. But that, if you look at the guidance, in Q6A on polymorphism, is that basically it's a requirement, it's an expectation that you will deliberately search for polymorphs. You need to not only monitor the batches that you produce and use throughout your development cycle, but you also need to deliberately try to vary the solvents, the compositions, the crystallization conditions in order to find as many forms of your drug as possible, and then use good science to establish which form is best for your product. And so that's where, and they also say this is a subset of the common techniques that are used. X-ray powder diffraction's kind of been considered to be the gold standard for identification of solid forms of drugs. Differential scanning calorimetry and thermogravimetric analysis is widely employed. Microscopy, a much underappreciated but extremely powerful technique, is also one that's used, and a variety of molecular spectroscopies are used.
Single crystal diffraction is just not used?
It is. It's used. It's used with. It's not generally does a drug product get produced in a form that's So the question was, is single crystal diffraction used commonly? In the screening phase, it's not commonly used, probably because of the expense of the technique. And in my lab, because we grew most of the discovery chemistry ultimately gave us, although I kind of worked at the discovery interface, but they gave us, because we grew most of the single crystals, we acquired the single crystal diffractometer and did all the structural analysis. And there are some ways, I'm not going to get into it here, but it's actually quite advantageous because you can calculate your powder diffraction patterns from your single crystal and firmly establish whether or not there's a mixture. So there's actually a really nice interplay between single crystal and powder diffraction, but it's generally not the final product, and the APIs are generally of smaller particle size. So it's not used as a traditional method for distinguishing polymorphs for the screening part.
So is polymorphic screening be part of NDA submission, or is it required?
Yeah, it's actually a requirement. So yes, the polymorph screening is an expectation and is a part of your regulatory document. You have to explain what all you've done and basically convince the FDA that you've done good science, and it ultimately factors into the control of your product, your API, as well as your product. And so they look for that in your regulatory submission, and hopefully when they see a robust package and rationale, they understand that you have good control of your process and product. And so ultimately, and you can actually, I think there's supposed to be 2 identification techniques. Ideally, you'll produce out of your API, once you've identified the forms and understand their thermodynamic relationships, you can get a process that'll produce a single form. And then you'll use a qualitative technique, oftentimes X-ray powder diffraction. You can substitute it. I think usually you have to have 2 analytical methods for identification of your API, and you can X-ray powder diffraction is one of those. Alternatively, it can be FTIR, which is a little less discerning from a crystallographic standpoint, but powder diffraction works very well for this. And so you can, ideally you have a single form present and it stays that single form. And if you have multiple forms, you might have it where, you look for these peaks that prove that you have the right form, and then you ensure at this position and this position and this position that this other form that's known to potentially come out of your process or that's relevant to your process isn't present. And so it's just a qualitative method where you just identify the single polymorphic form. But there are other special occasions where you might actually have a quantitative method developed. And I've done that on a few processes for specific reasons. And I'm not going to get into them here, but generally you try to get into where you have a qualitative method for identification of the form of the final product, API, and what's present in which the next slide goes to that though, is that you monitor during your stability, your accelerated stability testing, to ensure that there's no interconversions of forms and make sure that the form stays the same. If it doesn't, you'll end up having an acceptance criteria which will set specifications or limits as to how much is acceptable of an extraneous or different polymorphic form. But you'll put in your analytical tests, which high percentage of the time it'll be IR or Raman, most often though IR or X-ray powder diffraction to control your product. This is what I had within my lab, a whole variety of techniques, but certain ones are the most common, in particular for the screening. The conventional X-ray powder diffraction. Solid state NMR is extremely useful, higher resolution than the other molecular spectroscopies. But each of these have, it's nice to have multiple, they call them orthogonal techniques. Sometimes people refer to them as orthogonal techniques. I think it sounds nice, but basically different techniques are more sensitive to different types of forms. Oftentimes your thermal techniques are perhaps more, or even moisture absorption analysis might be more better at detecting small amounts of amorphous material within the presence of a crystalline, predominantly crystalline sample. But basically the different techniques, they serve in a complementary fashion. Microscopy, polarized light microscopy is extremely powerful, in particular when you couple it with hot stage microscopy. But to be honest, there's fewer practitioners that are very good at this. But you can learn a lot just even if you're not, even if you're not really experienced with the thermal microscopy or with microscopy, just by looking at a sample under, after you've done some crystallizations, if it looks hair-like, chances are it's not going to flow very well. So a lot of times by microscopy, I can tell you something that I might not be able to tell you that it will flow well, but I can tell you if it won't flow well a lot of times. And so you can quickly eliminate. So that's one of the things, and I find a lot of labs don't do this, and I've looked at a lot of data packages from different laboratories, and a lot of times they don't even, some labs don't even put microscopy or images in their data packages. And then later when you see the material that they're producing, you say, yeah, that stuff, your formulators will have a terrible time. They're going to kill you for selecting that form for a product. And it's just a 2-minute, it's the simplest technique, but it tells you a lot. And I encourage people highly to, and if you have a contractor that is doing polymorph studies for you, and if they don't have images of the form that they select for development, make them put a picture in, make them give you an image, look at it and see whether or not it's hairy or if it's got a nice morphology that might be, if not milled, if it's not equant, or if it doesn't look like it would be flowable, have them, make sure that there's enough three-dimensionality to it that it, or have them look until they find something that has an external morphology that looks like it might flow. Because I've dealt with this before, and it can be very challenging for your formulators. And it can cost a project a year or 2 in development. And we used high throughput screening, and then computational tools, a variety of computational tools. We had absorption modeling within our teams, but also, a lot of times it's you work hand in hand with your ADME and toxicology groups early on, and actually throughout the development, though, with the absorption modeling. So then I guess the third thing is so even if you have a form change during this period, it's not a crisis. It's nice, the sooner you have it, the sooner you can establish your doses. Elsewise you're going to have to see whether or not a form change makes a difference. And often one good place to do it would be during the bridging studies, usually when you go from that drug and capsule or one of those simple formulations. Usually you'll do, you'll have to bridge it to the commercial formulation to ensure that the bioavailability doesn't change substantially. And so at the same time you can actually, that's a nice time to switch polymorphs, but realize you're varying 2 things simultaneously. And so it's not ideal. It's nice if you have your form identified, your physical form identified early, so that this bridging study, there's only a single one involving the change to the commercial formulation for your pivotal studies for commercialization. But you can combine them too. That's a nice point to do a polymorph change if you need to. So the first system that I'll talk about is a colorful one that got around for a while. It was called Roy initially when I started on it. It was a intermediate for a drug called Zyprexa that sold widely at Lilly. That got up to over $4 billion a year, antipsychotic. But it was quite interesting because we had Eastman Kodak was producing the compound for us, the intermediate, and 1 minute it'd be red, the next time it'd be orange, the next time it would be yellow. They didn't understand. And classic polymorphism, so they dissolve it, solution in MR, and it was the same material each time. So classic polymorph, true polymorph. And it's a good example because visually, this is one of those few cases where polymorphism, you can't overlook the fact that this is a polymorphic material. It will form plates. It's the same chemical entity in different arrangements within the crystal lattice. So we did do the crystal structures of all of these forms, and the arrangement of the molecules were different. And also, the color was associated with the planarity of the aromatic rings. But it shows you very well how different substances can be just based upon the packing within the crystal lattice. And so, and you can envision also, if you saw something like this or this, it would flow a lot differently if this were a product. This is an intermediate, fortunately, but if this had been a product, these would likely flow much better as a powder if you micronized it down than these needles. These needles aren't terrible in my opinion, but sometimes you'll see stuff that's hairs that there's no way a formulator would be. If had this been a final product, these would have probably been more challenging from a flow perspective than these. And you could have told that very quickly just by looking under the microscope. But you look at the melting point differences, they're significant among these different forms. So this is a classic case of polymorphism, and it led to some very good science by one of my fine, with our group, and then with one of my colleagues. I had done some, just with high throughput screening from the early 2000s, just showed where multiple crystals would crystallize simultaneously from the same exact crystallization, and there was little control over what form would come out. You'd get mixtures of forms. A friend of mine who unfortunately passed this past year, Joel Bernstein, great scientist, and that's one of the wonderful things about this field. It's kind of a small field, and you have a few people, and then you all kind of come from similar lineages, and we all kind of connect internationally even. But Joel Bernstein was very prominent in the field, and he had termed this concomitant polymorphs, when multiple polymorphic forms grow simultaneously. Leanne Yu, in a nice Accounts of Chemical Research in 2010, I believe it was a cover article, but he continued to do very good work and went on to University of Minnesota. One of his former graduate students is here. Wisconsin, I'm sorry. Yes, University of Wisconsin. And he continued to study this interesting system, and I think actually there's 9 or 10 forms by now, of which I think 7 we have crystal structures of. But at this time, he'd use a combination of thermal analysis at the melting points and their enthalpies of fusion to, relative to the room temperature, the yellow form, which was stable at room temperature, he could infer the free energy, or measure the energy difference between the different polymorphs. And then by melting with the same miscible substance, he could measure the enthalpy of fusion and the melting point depression, and use the heat capacity changes to, again, at different temperatures with different amounts of that eutectic forming substance, could calculate at different temperatures the energy difference relative to the room temperature stable form. And I took the liberty of extending that a little bit, or extending those lines down to room temperature to get the Gibbs free energy difference between those forms approximately, so that I could use that in comparison with some other data that had been plotted for polymorphic forms. So this nice little thing that Abu Sarajuddin did in Trends and Solubilities of Polymorphs that he published. He took 81 different polymorphic forms and ranked the solubility ratio of the metastable form versus the stable form as polymorphic pairs. And he took 81 different substances or different polymorphic pairs and plotted those. And he found actually the solubility, the metastable form was about 1.76 times more soluble than the stable form. So there's some scatter in the data, but the average was actually pretty tight. And I was using like the free energy difference that Leanne had measured and extrapolating it down to room temperature. This data was determined by solubility analysis. And you can use a Hoffman equation to a first approximation. It's actually activities here, but you can use solubilities to the first approximation. And so I could take the free energy differences from the Roy system based on Leanne's data, and then calculate what that would be, that solubility ratio, and then compare it to the average polymorphic system. So the average polymorphic system, if you calculate based on this number, it's actually, I converted it into kcal instead of what Leanne had, which was the kilojoules per mole. But it's 0.33 kcal per mole is on average the difference for these polymorphic pairs. But with these, with the Roy compound, it's actually much tighter and quite a bit lower. So it was 0.14 kcal per mole, which is significantly different and pretty tight, and probably explains a little bit, I guess, the conclusion I had or inference I have is that, and I think there's enough data out there to, it's basically that polymorphs have very similar energy, in particular with those concomitant ones. There's competition of nucleation rate and growth rates that result in mixtures of polymorphic forms, and they're very difficult to control. If you want to control what polymorphic form comes out, you have to control the supersaturation ratios very tightly in order to do so. And really with Roy, it was pretty difficult to, because they were so tight, it was pretty difficult to, we were fortunate that it wasn't the final product, because it would have been a little bit, it would have been a challenging system. We might have ended up having to use some sort of, or, a quantitative method if it had been a final product. But it was much more, it was a difficult system to control what form came out.
How many percentage is amorphous versus crystalline?
That is not on here. That's actually in a different paper by, and I referenced it earlier, but it's as much as 1,000 times.
So amorphous is
Way out there, yeah. Yes, yeah. It's, but I did have, there's a reference in there, which fortunately was done by, or Bruno Hancock has one, but it can be up to 1,000 times different, yeah. And that's why people for the longest time were very hesitant to go with amorphous forms when a crystalline form was known, was because there was so much driving force or energy. And then we got into solid dispersions where you could inhibit that from happening. But I'm focusing on, the polymorphic systems, and I'll go, ritonavir's kind of been known over the years as one of the more famous cases with Abbott. They had a form that appeared after they had been on the market for a year or two with their semi-solid capsules, and all of a sudden a new form started to appear in their product. So a new crystal form was found in the formulated product. A team of scientists went to investigate if any significant changes had been made in their manufacturing process. Until this time, no detectable quantities of Form 2 had been detected in the bulk drug lots. Soon after visiting the manufacturing facility, Form 2 started showing up in their bulk drug production facilities as well. So they moved the seeds from the product, and then they seeded their process, and then all of a sudden they had trouble controlling their final process. They were off the market, I believe. This was first in class, I believe, of one of the protease inhibitors, and obviously there was a lot of, it was growing in sales very rapidly, and all of a sudden they had to withdraw from the market while they reformulated. They theorized that the compound adopted an energetically disfavorable conformation in the stable crystal structure, and that's why it didn't nucleate very well. To be honest, the data wasn't really good behind this. I mean, it was kind of a little hand waving, which is kind of what you find with a lot of times with these polymorphic forms. Didn't know, I mean, they theorized, but there wasn't an absolute answer as to what was going on. But they theorized that a heterogeneous nucleation occurred by a related substance that had that unfavorable conformation, nucleated it. This was by Bauer, who was in a different area, but at the same time, though, the guys in process chemistry published a paper, and they said the origin of Form 2 remains debatable. The fact was that this issue had to be addressed as soon as possible. And so there was a lot of urgency, and I knew enough of the guys from around there to know that, yeah, they were working a lot of hours. And what was interesting, though, is if you took it, it was a part of this study that Sarah June had done, and it was at one of the ones that's at extreme of differences in solubility ratio, and it was, if you calculated, it was about 0.8 kcals per mole. Theoretically, with polymorphs, they say that polymorphs will fall within 1 kcal per mole of one another. But the reality is most of them fall within 0.33 kcal per mole. So that brings me to my final system that is somewhat extraordinary. And so I called it the missing polymorph of melatonin agonist. But basically, if you remember Pasteur's experiments where he kind of led to some of our increased understanding of chirality and using tartaric acid, where he noticed under the microscope that there were half of the crystallites were of mirror image to the other half, non-superimposable, and it helped him infer, basically that they had, chirality associated with the chemical entity of tartaric acid. But in theory, if you took D and L tartaric acid and crystallized them separately, you could get the same forms out of each enantiomer. It's a mirror image, so they'd be mirror image crystals, but theoretically you could get the exact same packing out of them because there's the same molecules, they have the same conformational freedom and flexibility, they should produce the same forms. But in this case, the active enantiomer was produced by Discovery Chemistry. They decided that they would use this was in the mid to early 90s, and they went with the active enantiomer and provided for, and I've had this happen more than once, where to conserve the active molecule, we know biological systems are very sensitive to chirality. Crystallization, usually you do an achiral or non-chiral media, and so theoretically you should be able to learn what you need to from the inactive enantiomer. And so they, in the study of the inactive enantiomer, while they were off doing their tox studies and the like, 3 out of 4 of the initial batches that were provided were metastable form 1. The 4th batch resulted in this much more dramatically stable form 2. It was extremely difficult to produce this metastable form after the discovery of the stable form 2. And in fact, I only was able to go back and produce the metastable form 1 time. Not surprisingly that it came out of that it was from the oil, where I was actually able to get it to nucleate out as form 1. But it was much more difficult to get form 1 once the more stable form was produced. And that's not all that uncommon. I've seen it a few different times where you'd produce one form and then a next form and then a next form, and then ultimately you couldn't go back to the prior forms. It's been, Joel Bernstein again referred to that as being disappearing polymorphs, but it's been noted over history that this is not uncommon. There's a number of good papers where this has occurred. And some people say with ranitidine hydrochloride, which had a big patent dispute over it, but that was the case with ranitidine hydrochloride. So I had a disappearing polymorph in that case with the inactive enantiomer. But when I got the active enantiomer to work with, we never could reproduce the stable polymorphic form 2 that we knew existed from the inactive enantiomer. Even though we did thousands of recrystallization experiments and more than 10 multi-kilogram scale lots, each one of those produced the metastable form 1, which was quite surprising. So in that case, I called that the missing polymorph because we knew it had to exist, but we couldn't produce it. So what happens sometimes is you run into a big problem, and sometimes attrition takes care of the problem for you. And that molecule, we stopped developing it. It was in phase 2 clinical trials, I believe. We stopped developing it, but it took, we actually out-licensed it, and then finally the company that was actually working with it gave us, after the patent had expired, I got approval from on the chemical entity on legal to go ahead and actually the company that had purchased it from us went out of business, and then legal finally said I could publish on it. And so I had to wait 14 years, but ultimately if you wait long enough, you can publish it.
Good question on this. So when you have enantiomers like this, normally the racemates have a much lower energy, and the racemate's the first one to crystallize out.
But these were purified. So they actually, the very final step was a chiral producing step. Yes.
But if what you had observed there is correct, then it should be self-resolving, no?
Well, that's interesting. And I'm not going to actually be able to go into that in detail, but yeah, you would think that like Pasteur's experiment, that it would be a conglomerate. But it
should be so if you put the racemate in, then it should self-resolve because one has a much higher or a much lower.
We won't have time enough to go into that here, but there's some, if you have interest in this kind of thing, read the paper. I went into a lot, probably more detail than I should have, but it's an interesting, really interesting system. But so after 14 years, I thought, my mind, that really wasn't that way, was it? And, my mind was just, there's no way in the world it would still behave that way. But it was one of those that just stuck in my mind. And so once I got the approval, the first thing I did was actually, I got out of research stores, I got like 30 some lots of the active enantiomer and looked at them all, and they had all stayed stable, and they had stayed in that metastable crystalline form for 14 years, and I was pretty surprised with that. And then I looked at so then I took the 2 primary lots, one of the active enantiomer and one of the inactive enantiomer, and then I just recrystallized them against, I believe it was like 23 different solvents as separate experiments. And surprisingly enough, the active enantiomer melted or formed the metastable form, the one that melted 127.9 degrees C each time. And the inactive enantiomer always crystallized out at as a higher melting point in crystalline form. I didn't expect this to happen. I thought it was, I was amazed that would occur.
So is these results published or?
Yeah, I did. Yeah, I'll reference it in here, but.
What I learned is 2 racemic enantiomer here. The physical property should be the same.
They should. You're absolutely right.
What I learned is the
Yeah. You would think they would be, but they weren't. And so I studied it, and I'm not going to go into all the details, but but part of it was, obviously, related substances being most similar to the drug itself are the most likely culprit. And but in this case, the purity was almost identical. They're both, one's 99.3% pure, the other's 99.5%. They had the same related substance profile, just slight, very small differences in the rel subs. Nucleation is what I'm going to say is very interesting, and we still don't fully understand it. I was able to take the 2 individual forms and make a racemate, but I did a lot of other studies that are published here trying to understand it.
It crystallizes the racemate.
It does crystallize as a racemate. Yes, it does. Yeah. Yeah, and so that actually makes some pretty nice cover art. So this is if you actually do the experimental, I should have left the experimental points on there probably, but it doesn't look as pretty if you do. But I measured the actual thermal phase diagram. But so in the one case, because it's the lower melting point, when I did the melt eutectic, it had a different eutectic because it was a different polymorph mixed in with the racemic. And when I did but your lower melting point form has a polymorph that's obviously missing because we know that it should form that polymorph. But in the other hand, we had with the inactive, once you formed the stable polymorph, the one that melted so much higher, you couldn't form other than the one time where I did it from the melt, or from viscous liquid, supersaturated liquid, was able to get it. But essentially you had a disappeared polymorph. It had become much more difficult to form that metastable form. And so I did publish it, a couple of, there's some, but this is the primary paper, but there's a lot of detail in there because I'd hope somebody would go back and try to reproduce the work for themselves and actually find that missing polymorph. I used a variety of techniques, but I did want to relate again through solubility and through calorimetry, the phase diagrams, and also the solubility analysis. To get to the main point, basically, is that it was almost like ritonavir also, where that metastable form was fourfold times more soluble than the stable polymorph. So it was pretty exceptional. So these ones that are really hard to nucleate oftentimes are the ones that have the highest difference in energy. The ones that nucleate the most, I'll get into that in a second, the ones that nucleate more readily, or the first appearing ones, are the ones that are metastable, and that's been known for years. But basically, melatonin and ritonavir, the late appearing polymorphs, or the difficult appearing polymorphs, if you will, were different by 0.8 kcals per mole. So they are at the extreme for polymorphs. Roy was 0.14 kcal per mole. That's the concomitant one where we had difficult controlling which form would grow. So late appearing polymorphs are nearly 4 standard deviations away from the average energy difference of the average organic polymorphic systems. And so I did do these energy diagrams using the combination of thermal analysis and solubility analysis across a wide variety of energies, or of temperatures. But I'm just going to boil it down to, when it comes to polymorphism, it's all about nucleation and getting different conditions and getting forms to nucleate. And the classic nucleation theory, which there's other, nucleation theories out there, density fluctuations and the like, but to be honest, it's still one of the most practical descriptions of nucleation processes. And but typically you'll break down the energy of the nucleation process into 2 separate terms. The initial term that is disfavorable is associated with interfacial tension. And then there's this other term which is associated with volume growth of the actual crystal. And so that interfacial energy term is disfavorable, and that's because so right here, as the radius of clusters of molecules, as they come together and form larger and larger clusters, it's disfavored as they separate themselves from solution. There's an interfacial tension that occurs. But then once you get enough molecules together beyond that, the bulk growth becomes dominant and further growth and the crystallization, the overall crystallization process becomes favorable and crystallization occurs. But some people say primary nucleation doesn't exist. I've heard that before from some pretty good scientists, but secondary nucleation, whether you consider it by seeds, which is something we use to our advantage in processes, lots of people, it'd be preferable that you wouldn't have to use seeds, but it's your best way to control crystallization of a process. And it's always prudent to write in a seeding step. You might not have to use it, but write in provisions for a seeding step in your final crystallizations for control purposes. But if you remove that interfacial tension through seeding, you can direct the crystallization to the form that you desire. And so if you have seed crystals, you bypass the disfavored process of nucleation. And so if you do the inverse of that, which is nucleation rate, basically the bigger the energy difference between the crystalline forms, the higher basically this nucleation activation energy. And so your metastable forms tend to nucleate at a faster rate than your stable crystalline forms. And so in the end, in the absence of seeds, when the energy difference between polymorphs are small, their nucleation rates are likely to be similar, and concomitant polymorphs more likely. In the absence of seeds, very large energy differences may result in very large differences in nucleation rates. And so the overall conclusions, this is kind of a reiteration of the earlier thing, but for the melatonin agonist, my inference is that there was basically a heterogeneous impurity, likely not the drug substance itself, because both of them were exposed to the same, the same environment. They were grown in the same labs with the same equipment. But it's likely that a chiral impurity enabled one enantiomer, heterogeneously nucleated one, whereas the other one was never exposed to that same mirror image chiral substance that caused heterogeneous nucleation to occur and crystallization of its stable form. But with that, I'd like to acknowledge my coworkers, Craig Wolfangle, Jennifer Runyon, and Dave Jackson for their analytical support, Professor Frank Lucien, who passed away, unfortunately, way too young, and John Kendrick, who worked with me on the ab initio calculations, which I didn't present today, but are in the paper. Thank you for your time and listening to my presentation. That's pharmaformfinders.com. Is the company that I work for as an independent consultant. And I thank very much Regis Chemicals for giving me the opportunity to speak today.
Anybody have any questions?
I've got a question.
Can I get her first? She was up quicker.
So I just had a quick question, kind of on the last point that you were making about there being a chiral impurity that you think was doing this. And so this is the chromatogram you have here. I don't know what's that. Is this the chiral method? Is there any?
You're right. This is not a chiral method.
Okay. No?
They would resolve.
Yeah.
Yeah. No, at the time we were doing it, but when I went back and visited, I did not have access to the resources, so I didn't have that data to include. Yes, I would have liked to have had that.
Yeah. Okay. That seems to be kind of the missing thing.
It is. Although it should have the same chromophore, right? But one of those in the same entities were present there. But yes, that would be worth knowing if one of those were chiral. I should have. Yeah, it's been a while. So, thank you.
I find it interesting that you said that a typical anhydrate versus hydrate, the anhydrate has a 4x better solubility than the anhydrate. Does that imply that the new polymorph is different? Are they interconvertible by a DVS, for example, with the
Yeah.
curve? Or yes or no?
Yes.
They would be interconvertible?
So anhydrate, sometimes, it depends on the nucleation, how well it nucleates. So the question was, would anhydrate and hydrate if you're doing the vapor sorption, desorption, so changing the water activity in that piece of equipment. I find that if it's very soluble, the interconversion can oftentimes be absorbed or be observed in the vapor sorption experiment. Sometimes I think if you really want to be safe, you're better off putting it into, because it's an expensive piece of equipment and it takes a long time to make the measurement, or you're tying up a piece of equipment, it may take a while for it to nucleate the hydrate. 40%, 30 to 40% of the molecules will form a hydrated form. It's been shown at least in 3 or 4 good publications. And they always come up with about the same, the 30 to 40% of the time. So you should almost always expect that you're going to find a hydrate. You want to choose the form that is stable from, say, 30 to 70% relative humidity. It's nice if it doesn't interconvert. But to really be certain of whether or not a hydrate exists, DVS is extremely useful for understanding hygroscopicity, and whether or not it happens quickly changes from one form to another. But it's also nice to use, to place the sample over water at different water activities.
Right. What I'm getting at is sometimes you come up with let's say I did single crystal diffraction on the both, and it was essentially the same. It's just the water's there in one and not the other. Is that a different case than some hydrates where it's a different?
It is different.
Which is more typical?
I'm not going to say either of them is more typical. I've experienced probably half the time it's one where, and we oftentimes refer to those. So the question was, there's certain hydrates where the water molecule is an intimate part of the structure, and if you remove it, the structure will collapse. And there's other cases where you can remove the water by exposing it to low humidity or atmospheres of low water activity. And in those cases, usually there's a change in the lattice. It can be very small, but usually there's a change in the lattice, and usually there's actually fracture, so you can see it under microscopy. But sometimes the peaks stay almost exactly the same location, so you don't get a collapse of the structure. And those we call variable hydrates, and usually when we're doing our screening process, we try to stay away from those because it's more of a challenge analytically and other ways of controlling the potency, because you got to include water in the potency. So usually we try to stay away from those types, from selecting those as final products. But your cephalexin, which was called monohydrate, and then they made us change it to cephalexin hydrate because same thing with C-chlor, lots of molecules do that variable hydrate sort of behavior. It's just a little more challenging. Sometimes you have no choice, it's the only form you have, but other times, yeah. But I wouldn't say one's more common than the other. Run into them both.
A distinct difference in reversibility during TBS. Is that a sign, a difference between, say, adventitious water absorption? Is that a sign maybe you're seeing an anhydrate that has such a distinct physical property that it changes?
I'd say you're probably right. If you have a nice distinct step, usually that's a physical form change, as opposed to like with cephalexin, or it changes kind of, you get hysteresis, but it's not a real discrete change in the DVS.
But absence of that may not mean anything, but if you see it, it could be a strong signal.
Usually it's a good, that means it's a nice discrete change. The only thing is if you have hysteresis between them, it's a nice step, and you have hysteresis between them. It's, you know that there's an interconversion, you just don't know where in that humidity range that occurs. But that's when we go on to slurries and solvents with water and determine what that critical water activity is to pin it down closer. So, okay. Thank you. Yes.
If you found a more stable form is because present related impurity, are you still going to pursue that more stable form or not? Or just
Yeah.
I need to present on this one,
Oh.
this amount.
So the question is, if you know that form is more stable, but you can't get it, are you going to market the metastable form because you can't get it? You know, I think that's what you would do if you had to, but I can tell you that if we had gone beyond phase 2 clinical trials, I can't imagine how many people in our company, we had a significant number of people working on it, but we would have had all the king's horses and all the king's men trying to find that polymorph. I promise you. Because somebody else could have patented that other form, and we couldn't make that form, so there's no way we could patent it.
In that situation, is it better to pursue the amorphous, like a square line?
Possibly.
Just to take that out of the equation.
That
Never
possible.
really goes out of the equation, but you know what I mean?
You know, that I'm sure we would have considered that. Yes, it actually happened before people really went to doing more of those, but yeah, that would have been a good option.
So if you're young, phase 3, you then with phase 3 study in the metastable form, and then you do the bridging study, show that because the solubility, you don't need a high solubility, maybe they are equivalent. Is that sufficient to justify? I don't need to repeat this phase 3
Yeah,
study.
you'll have to go through the ICH guidelines. Yes, you can go with metastable forms. There have been plenty of metastable forms that have been marketed, and then they've later found stable forms. But as long as they can continue to produce the same form, even if it's a metastable form, there's nothing that precludes you from developing that metastable form. But there's a risk.
Develop a metastable form. I said if you done with the pivotal study of metastable form, you found more stable form, but you already done with the pivotal study, and you just do a bridging study. Do you need to go back to repeat those pivotal study? You can say, well, that's equivalency. So
I think that's good science, and that'd be dialogue between you and the FDA, or you and the regulatory body. But you could rationalize through modeling and other things whether or not.
So, I have a question on your two enantiomers that you crystallized out. So, did you have the single crystal for each of these enantiomers? So, was there a conformation? So, if you think about, there's an amide bond in there,
Yeah.
right? So, and you have a different chiral center.
Uh-huh.
So, you could potentially envision a situation where you have a chiral center, and then your amide bond, the way it sits, imparts sort of like an atropisomerism type
Yeah,
thing.
yeah,
And
yeah.
you end up with two different diastereomers now. So they're no longer enantiomers, but what you have are the equivalent of an atrodiastereomer.
You
Is
could.
that possible? Or is
I
that
mean,
the
you
reason
could
why-
rationalize that, but that would be available to either one of them.
Maybe not, because that's where so in one of the enantiomers, where potentially the other bond rotates and sits could be affected.
Well, okay, yeah, you're saying, yeah,
So that
it's
could- that could
possible.
cause essentially like an atropisomerism.
If it's a high enough energy barrier. I don't
Correct,
think in
between
this
the
case,
conversion of the two.
I don't think in this case you have a high enough energy barrier that, I mean, those are single bonds. I doubt it, but I read the paper, think about it, send me an
It
email.
looks really curious.
It is, it's a unique.
I think
Unique.
in your crystallization system, there must be some chiral effect. You didn't find, but other than that, there's no explanation why one form is
Yeah.
from one or the other from two.
Yeah,
Maybe something
and I think that's,
minor things.
polymorphism's unique, and nucleation's still fascinating today as it was 100 years ago.
Yeah, and from your solubility, you're saying it's about 1.3 sorry, 0.8 gigaounce.
Yeah,
So
point, or 4-fold.
conformationally, 0.8 gigaounce is not a whole heck of a lot.
No, no.
It's like that.
No,
that, right?
but I don't think that's your explanation, but anyway, if there's any other questions, if not, I'll let you guys eat.